Hi, in today's biochemistry bits, we are going to discuss a reasoning out question. So the question is hemolysis in viper bite or poisoning. This is a very common question asked under the topic of chemistry of lipids for a first MBBS student. So what is this viper? Viper bite or poisoning means. So vipers are a group of or a family of snakes found across different continents across the world, and the venom or the toxin which is produced by this viper snake is considered to be hemotoxic, or it can cause hemolysis. So we are here to see why there is hemolysis in this viper poisoning. The basic concept which needs to be understood to tell the answer for why there is hemolysis in viper poisoning is about phospholipids. Okay, what are phospholipids? Every one of you would have known that phospholipid bilayer is a component of all the cell membranes. Okay, and it is arranged like this, a bilayer. Inside you will have a tail which is hydrophobic, and outside you have a head which is hydrophilic, which is the extracellular part, and this is the intracellular part. Okay, so this is how this phospholipid bilayer is arranged in our cell membranes. So if you zoom this and see, you will be able to see this particular arrangement of the or the chemistry or the structure of a phospholipid. Okay, so this is the overview, and inside if you zoom in, how the tail is and how the head is shown clearly here. So phospholipids will be divided into two groups, glycerophospholipids or sphingophospholipids, based upon the type of alcohol. So here we have the glycerol backbone. You all know the structure of glycerol. You have three carbons with three OH groups arranged. Okay, CH2OH, CHOH and CH2OH. Here, each OH will be replaced by a different constituent. Okay, so first OH group, that is, this is the first carbon. It is replaced by a fatty acid, and it is a saturated fatty acid which is represented here. Second carbon of glycerol is also replaced, in that OH is also replaced by another fatty acid. This is a unsaturated fatty acid which is having a double bond, so it has a kink in this structure. So this is an unsaturated fatty acid. Third carbon is attached to a phosphate group and an R group. Depending upon the R group, you name the phospholipid. The most common phospholipid in cell membranes is lecithin, where the R group will be a choline. Okay, choline will be replaced in the R group, and that is called phosphatidylcholine or lecithin, which is the commonest phospholipid. Now we are here to see the various types of phospholipases which acts on the phospholipids. So this image shows four types of phospholipases and its site of action. Phospholipase A1, phospholipase A2, phospholipase C, phospholipase D. Okay. Phospholipase A1. We told in glycerol there are three carbons, right? That is numbered in this particular place. 1, 2 and 3 carbons are numbered. Okay. The first carbon is attached to a saturated fatty acid commonly. So if that place is being cut, it is by phospholipase A1. If the second fatty acid, that is unsaturated fatty acid most commonly, if that fatty acid is being cut, it is by the enzyme phospholipase A2. Then phospholipase C will cut at the phosphate joining point. Okay. And phospholipase D will cut at the special group which is going to differentiate the different types of phospholipids. This is the R group which we have been seeing in the previous slide. So these are the sites of action of various phospholipases. Among this, we are more concerned about the phospholipase A2. Now let's see the reason for hemolysis in viper poisoning. So the viper venom or the viper toxin will contain the enzyme phospholipase A2. You remember the site of action? Phospholipase A2 will cut at the second unsaturated fatty acid which is attached to the glycerol backbone. So when it acts on particular phospholipids, most commonly which is lecithin on the present in the RBC membrane, this phospholipase A2 will act on these phospholipids which are present in the RBC membrane. When it acts on lecithin, the products formed are lysolecithin and the unsaturated fatty acid. Okay, this lysolecithin is the culprit agent which is going to cause the hemolysis. Lysolecithin has detergent action and it can destroy the RBC cell membrane, leading to hemolysis.